gene therapy. So in that, CAR T cell therapy. CAR T cell therapy. So gene therapy is a medical approach which is used to treat the genetic abnormalities, genetic problems by correcting the underlying genetic problems. So medical approach, that means gene therapy is a medical approach to treat the diseases by correcting the underlying genetic abnormalities, correcting underlying genetic problems. That we call as a gene therapy. That means the abnormal genes can be rectified. Say for example, the CAR T cell therapy. So this CAR T cell therapy, it is a cell-based gene therapy. It is called as cell-based gene therapy. Recently, the scientists have developed a gene therapy called as CAR T cell therapy. Here CAR represents chimeric antigen receptor T cell therapy. T cell means T lymphocyte. So scientists from US have developed a new gene therapy that is called as just CAR T cell therapy. CAR T cell therapy, chimeric antigen receptor T cell therapy. So what is chimeric antigen receptor? Suppose say this is T cell. Say this is T cell. First result is T cell, and this is a antigen receptor. This is a antigen receptor. Here, by this gene therapy, the scientists have modified changed this antigenic receptor. That means the gene responsible for this, the DNA responsible for this antigenic receptor have been altered, changed, in order to work this antigenic receptor 24/7 continuously. That means it is acting as a live drug. That means what is that antigenic receptor? This antigenic receptor will identify the incoming antigen. The antigenic receptor will identify the incoming antigen and gives the information of this antigen to the T cell. Thereby, the T cell, with the help of B lymphocyte, releases the antibodies against to this antigen. Are you getting my point? Whenever an antigen enters into our body, so actually, see, this is T cell. This is T cell. It is having antigenic receptor. Antigenic receptor. Antigenic receptor means its function is to identify the incoming antigen. Simply, its function is to identify the incoming antigen. And this information will be given to the T lymphocyte. T lymphocyte, with the help of B lymphocyte cell, releases the antibodies against to this antigen. And these antibodies will fight with the antigen that will be killed. Like that, it protects our body. This is called cell-mediated immune response. Is it clear? In this CAR T cell therapy, the scientists have altered this antigenic receptor. Chimeric antigenic receptor. That means the DNA responsible for this antigenic receptor have been modified in such a way that it works 24/7. It continuously identifies the antigen responsible for the cancer. Got it? So whenever the antigen responsible for the cancer comes here, immediately this antigen receptor identifies, it triggers this T cell, thereby T cell, with the help of B cell, releases the antibody against this cancer. So in any time, if antigenic, that means the antigen responsible for cancer enters, So the antibody will be released immediately, that will be killed, that kills this antigen. Like that, the antibodies related to this cancer are continuously produced within the lab, within the body itself. That means it is acting as a live drug. This type of gene therapy have been developed by the scientists, and it has been approved by US FDA also. Okay, US Food Drug and Administration. This also approved this one, and this is called as a Yescarta therapy. Yescarta therapy. You remember this one? The scientists from US have developed a gene therapy which is used to treat the cancers. That means this CAR T cell therapy, it acts as live drug. It releases the antibodies within the body against the cancer. Okay, this is also called as Yescarta therapy. Yescarta therapy. Simply you remember here, CAR T cell therapy. It is a cell-based gene therapy. Within the T lymphocyte cell, the genetic material is altered in order to work this antigenic receptor continuously. Okay, this is called as a CAR T cell therapy. You remember this abbreviation, that's enough. Chimeric antigen receptor T cell therapies. And this was approved by US FDA, United States Food Drug and Administration. This is also called as Yescarta therapy. Yescarta therapy. Right? And next one is gene editing. Gene editing. This was given in the previous exam. Gene editing by CRISPR-Cas9 technology. Gene editing by CRISPR-Cas9 technology. So this was given in the previous exam. So simple question was given, which of the following is a gene editing mechanism? He has given options: CAR T cell therapy, CRISPR-Cas9 mechanism, okay, PCR technique. So that is a rDNA technology. Like that, he has given options. So the answer was CRISPR-Cas9 technology. So gene editing or genome editing. So by this gene editing process, the scientists can able to alter the genetic material. Suppose say this is the DNA. Suppose say this is the DNA. This is the pattern of arrangement of nitrogen bases of the DNA. Suppose if this part of the DNA is modified, suppose if this part of the DNA is abnormal, if this part of the DNA is abnormal, if you want to change this DNA, if you want to alter, if you want to rectify this DNA, this can be, this method, CRISPR-Cas9 technology method is used. What is that CRISPR-Cas9? You have to remember this abbreviation. CRISPR. CRISPR means cluster. CRISPR, cluster, regularly interspaced, short, clustered, regularly, clustered, regularly, interspaced, short, palindromic repeats. Clustered, regularly, interspaced, short, palindromic repeats. You remember. Then Cas9. Cas9 means CRISPR associated protein. CRISPR associated protein. Cas9 means CRISPR associated protein. Right, now listen, just listen. So actually, you remember gene editing. This CRISPR-Cas9 technology is one method of gene editing mechanism. Here, what is CRISPR? Try to understand this one. Cluster regularly interspaced. Clustered means group of. Regularly interspaced. Short, small. Palindromic repeats. Palindromic repeats means mirror images, you can say. Okay, if you read it in forward and backward direction, they read same. Okay, they read same. So that means if you read it in forward as well as in the backward direction, it reads same. That is called as mirror images. These are called palindromic repeats. Suppose say for example, if you consider the DNA sequence, okay, GAA TTP, GAA TTC. Okay, so this is one sequence of nitrogen bases in the DNA: guanine, adenine, thymine, cytosine. If you read it in backward direction, complementarily, the complementarity of this one, C complementary is G, T complementary A, T complementary A complementary T, C. Got it? That means if you read it in forward direction, GAA TTC. Complementarily, if you read it in backward direction, same GAA TTC. And these type of sequences are called as palindromic repeats, palindromic sequences. And these palindromic sequences are present only in some prokaryotes, like in some bacteria. Got it? The palindromic repeats, palindromic sequences are seen in the bacteria only. By using these type of sequences, this sequence can be corrected. This sequence can be rectified without change in the sequence. Is it clear? And this mechanism is called gene editing mechanism. No need to go in depth of this mechanism. Just you have to understand the abbreviations of this one. So CRISPR-Cas9 technology is a one type of gene editing mechanism. So in which the scientists have used some palindromic repeats which are extracted from the prokaryotic organ, that is from the bacteria. And here Cas9 means CRISPR associated protein which is acting as endonuclease, which is acting as restriction enzyme. Is it clear? Right, that you have to remember. What is just gene editing mechanism? CRISPR-Cas9 technology. Okay, you have remember the abbreviation of this one. And next one is here is scientist from Oregon Health and Science University, US. Scientists from Oregon scientists from Oregon Health and Science University, Oregon Health and Science University from US. Scientists from Oregon Health and Science University, US, for the first time have successfully edited genes of human embryos. Scientists from Oregon Health and Science University, US, for the first time have successfully edited genes from human embryos. Have successfully edited genes from human embryos for correcting defective DNA. For correcting defective DNA. Right. Next point you write down. First gene edited babies. First gene edited babies. Lulu and Nana. First gene edited babies, Lulu and Nana. Right. In 2018, a Chinese doctor for the first time. In 2018, a Chinese doctor for the first time performed gene editing on embryonic stem cell, on embryonic stem cell, gene editing on embryonic stem cell using CRISPR-Cas9 technology. In 2018, for the first time, a Chinese doctor performed gene editing on human embryonic stem cells using CRISPR-Cas9 technology. The CRISPR-Cas9 technique. The CRISPR-Cas9 technique was used to modify the CCR5 gene. The CRISPR-Cas9 technique was used to modify the CCR5 gene on the embryonic cells of the couple, to make them resistant to HIV virus. They modify the CCR5 gene on the embryonic stem cells in order to resist against HIV virus. So Chinese doctor for the first time performed a gene editing mechanism by using CRISPR-Cas9 technology on the embryonic stem cells in order to resist the gene against HIV virus. Okay, right. So these couples gave birth to this gene edited couple gave birth to the babies called Lulu and Nana, twins, Lulu and Nana. Next. Right. So next one is DNA fingerprinting, or DNA from profiling, or it is also called a DNA testing. So next one is DNA fingerprinting or DNA profiling. DNA fingerprinting or DNA profiling or DNA testing. So creation of biometric database. Creation of biometric database of individuals' DNA. So creation of biometric database of individuals' DNA information. Individual DNA. That is called as a DNA fingerprinting. Now listen. Suppose if you consider the DNA, say this is DNA. This DNA is made up of nitrogen base, sugar and phosphate. You know it very well. So mainly the genetic information is stored in the nitrogen bases. And here what type of nitrogen bases are they? And how these nitrogen bases are arranged? What are the pattern of arrangement of these nitrogen bases? That can be known by this DNA fingerprinting or DNA analysis, or you can say DNA testing. So this is the creation of biometric database of individual information of individual's DNA. Is it clear? So that is called as DNA fingerprinting. Actually, if you consider the DNA sequence here, okay, if you consider the sequences may be of different types, like unique sequences, unique sequence, repetitive sequence, repetitive sequence, and highly repetitive sequence. Repetitive sequence, highly repetitive sequence. So unique sequence means what? They are unique, they are specific. Suppose some sequences are there, they are not repeated in anybody. They are unique to that particular person only. Okay. So that means if you consider the human DNA, 99.9% of DNA, 99.9% of human DNA is same. But 0.1% of DNA of an individual is unique. It is not repeated. Because of this 0.1% of DNA only we are getting unique characters. Okay. 99.9% of human DNA is similar. But 0.1% DNA is different. That 0.1% DNA makes the uniqueness of that particular person. Are you getting my point? Okay, these are called unique sequences. Okay, these unique sequences may varies from one person to other person. Are you getting? So these are called VNTRs. VNTRs means variable number tandem repeats. Variable number tandem repeats, VNTRs. The VNTRs varies from one person to other person. The VNTRs of, that means the frequency of repetition of VNTRs may be matching between the closely blood related individuals. Variable number tandem repeat. These are responsible for DNA fingerprinting. So, what is DNA fingerprinting? Creation of biometric database of individuals' DNA. Here, if you observe the DNA sequences, the sequences varies. They may be unique sequences, repetitive sequences, highly repetitive sequences. Unique sequences means they are unique to that particular person only. 99.9% of the DNA of human beings may be same, but 0.1% DNA is different. That makes the unique characteristic features of that particular person. So these unique sequences are called as VNTRs, variable number tandem repeats. So the VNTRs may varies from one person to other person, but they are similar. The frequency of repetition of these VNTRs may be matching between the closely blood related individuals. Based on that concept, the DNA fingerprinting is done. Is it clear? Right. Then what are the applications of this DNA fingerprinting? Applications of DNA fingerprinting. Applications of DNA fingerprinting. DNA fingerprinting is a laboratory technique. It is a laboratory technique used to establish a link between DNA fingerprinting is a laboratory technique used to establish a link between biological evidence and a suspect in a criminal investigation. So biological evidence and a suspect in a criminal investigation. So that means the DNA fingerprinting is used in criminal investigation process. The DNA fingerprinting is used in criminal investigation. That means this technique is used to establish a link between biological evidence and the suspect in the criminal investigation. How? That means a sample is collected from the scene of offense. Sample like a hair, blood smear, any cell, okay, tissue, saliva, etc. So semen, okay, these can be collected. from the scene of offense. And the DNA is extracted from this one. DNA is extracted. Okay, this DNA is extracted, and here an electrophotogram is made from this, like this. So this is the sample DNA. This is the sample DNA. And this DNA, this is the DNA collected from the suspect. Suspect 1, suspect 2. So that means a sample like hair, blood, cell, tissue, saliva, semen, whatever it may be, collected from the scene of offense. From these samples, the DNA is collected, DNA is extracted. This DNA can be matched with the suspects. Okay, say this is suspect 1, suspect 2. Suppose this is the electrophotogram of the, say this is the electrophotogram of the sample DNA. Okay, when you digest it, when you restrict it, you will get various fragments. These are the fragments of the sample DNA. When you compare these fragments with the suspect DNA, say this is suspect 1. So the fragments may be like this. They are not matched. Okay, if you consider the suspect 2, so these are the fragments from the suspect 2. Say these are the fragments from the, there is a matching. The matching is in between 70 to 90%. Okay, if minimum 70%, maximum 90% matching is there, then that can be considered as a, that suspect can be considered as a criminal. Is it clear? Like that, in criminal investigation process, the DNA fingerprinting process is involved by matching the suspect DNA with the sample DNA. When there is a match, that will be that is confirmed as a, he involved in the criminal, he involved in the crime. If it is not matched, the DNA sample is not from the suspect. Is it clear? So like that, the link between the biological evidence and the suspects can be established with the help of this DNA fingerprinting. And also the paternity can be established by using DNA fingerprinting. The parental disputes, by using this DNA fingerprinting, various types of mysteries like robberies, murders, rapes, as well as the parental disputes can be solved. So by using this DNA fingerprinting, the various types of mysteries like robberies, murders, rapes, even the parental disputes can be solved. So this is the importance of DNA fingerprinting. And also identify the that means identity of the persons can be maintained by this or to establish the identity of the persons, the DNA fingerprinting can be used. So these are the various techniques of DNA fingerprinting. Right. What's the next topic? GM crops. Okay. Right. So this biotechnology has vast applications in agriculture also. So this biotechnology can help GM crops. So this biotechnology has vast applications in agriculture. It can help meet ever increasing need by increasing the yields. So that means by modern biotechnology, we can insert a gene into an organism, that is into a crop, thereby we can get the desired characters. Otherwise, we can remove the unwanted characters from the crops. So these crops which are genetically modified, these are also called as transgenic crops. So what is transgenic crops? A gene of interest. Suppose say this is gene, or you can say this is DNA. A DNA of interest is inserted into a crop plant, thereby this crop becomes GM crop. Got it? So a gene of interest is inserted into the crop, thereby we can get a new crop that we call a genetically modified crop. What are the benefits of these GM crops? Increased yield or increased production. Increased yield or increased production. Improved nutritional value. Improved nutritional value. That means the nutritional value of the crops can be increased, and we can increase the shelf life. Increased shelf life of the crops, and also crops with environmental benefits. Crops with environmental benefits can be produced. Crops with environmental benefits. Increased yield or increased crop production, improved nutritional value, increased shelf life of the crops, and environmental benefits. Environmental benefits. Right. So let us see some important examples of these GM crops. So what are GM crops? Genetically modified crops, or they are also called as transgenic crops. A gene of interest, a part of the DNA or gene of interest can be introduced into the crop, thereby we can get genetically modified crops or transgenic crops. Transgenic crops. So these are the benefits of transgenic crops. Let us see some important examples of these transgenic crops or GM crops. The examples includes golden rice, flower sour tomato, golden rice, flower sour tomato, BT cotton, BT brinjal, BT cotton, BT brinjal, GM mustard, Arka Shubha, HTBT cotton, Arka Shubha, HTBT cotton, and also DMH11. These are some important examples of GM crops. Golden rice, flower sour tomato, BT cotton, BT brinjal, GM mustard, Arka Shubha, HTBT cotton, DMH11. These are some important examples of genetically modified crops. Over. Over. Right. So let us see one by one. So golden rice. So actually golden rice. So it is a GM, genetically modified rice, to contain high amounts of beta carotene. To contains high amounts of beta carotene. What is beta carotene? High amounts of beta carotene. Beta carotene means it is a precursor of vitamin A. It is the raw material of vitamin A. So golden rice it is a genetically modified rice variety to contain high amounts of beta carotene to address the night blindness problem. Beta carotene, it is to address night blindness. So what is night blindness? Unable to see the objects during night time. This is because of deficiency of vitamin A. That is rhodopsin production decreases. So improper production of rod cells. So due to the deficiency of vitamin A, I already told you, due to the deficiency of vitamin A, the rod cells will not function properly. So the rhodopsin production decreases. When the rhodopsin production decreases, we can't see the object during night time. That condition is called as a night blindness. In order to address this night blindness problem, the scientists have developed a genetically modified rice variety called as golden rice. So there is a rice variety called tapai rice variety. Tapai rice. So in this tapai rice variety, 3 genes have been introduced. 3 genes have been introduced. 2 genes from 2 genes from daffodils, 1 gene from bacteria. 3 genes have been introduced in order to produce high amounts of beta carotene. This is called as a golden rice. Just you remember, simply you remember what is golden rice. Golden rice is a rice variety containing high amounts of beta carotene. That is vitamin A. Next, flower sour tomato. Flower sour tomato. That is flower sour tomato. So that means it is a genetically modified tomato in which the ripening is delayed. Ripening is delayed. Flower sour tomato in which the ripening process is delayed by gene suppression process. Ripening is delayed by gene suppression. So I already told you, by this genetic modification, we can introduce our desired gene into the crop, or we can remove the unwanted gene. So that means we can enhance the functionality or we can suppress the functionality. Here in this flower sour tomato, what we are doing, the ripening is delayed, means we are suppressing the activity of the gene. If you observe the ripening process, suppose if you consider the tomato, if you consider this tomato, say this is tomato. Generally, say this is tomato. Here, the cell wall of this tomato, the cell wall of tomato contains pectin, a substance called pectin. Pectin is present. During ripening process, what happens? The pectin gets degraded by an enzyme called pectinase enzyme. So generally, the cell wall of tomato contains a substance called pectin, which is made up of pectin. By the activity of this pectinase enzyme, the pectin gets degraded. When start degrading, the cell wall becomes smooth. Okay, so the ripening process will be started. Are you getting my point? Okay, now this pectinase is indirectly activated by another enzyme called beta-galactosuronase. The activity of this pectinase is enhanced by another enzyme called beta-galactosuronase. Are you getting my point? Generally, tomato is there. The tomato contains cell wall. The cell wall of tomato contains a substance called pectin. And this pectin is dissolved by this enzyme called pectinase enzyme. When the pectinase enzyme starts functioning, the ripening process will be started. Is it clear? Indirectly, the activity of this pectinase enzyme is enhanced by another enzyme called beta-galactosuronase. Okay. Now, by this gene suppression process, we are suppressing the activity of beta-galactosuronase. By gene suppression process, we are suppressing the activity of beta-galactosuronase. When you suppress the activity of beta-galactosuronase, automatically the activity of pectinase will be reduced. అర్థమైందా లేదా? అర్థమైందా? Are you following? So when you suppress the activity of beta-galactorinase, indirectly we are suppressing the activity of pectinase. When the activity of pectinase is decreased, so the degradation of the pectin will be slow down. When it slow down means ripening process will be slow down or delayed. Is it clear? So this is flower sour tomato in which the ripening is delayed. In order to store for longer time, okay, in order to transport for longer distances, we have developed this flower sour tomato, right? This is a flower sour tomato. Another one is BT cotton. Right. So another one is BT cotton or BT brinjal. So both are one and same. BT cotton and BT brinjal. So here in BT cotton or in BT brinjal, BT stands for Bacillus thuringiensis. Bacillus thuringiensis. It is a soil bacteria. Bacillus thuringiensis is a soil bacteria. This is soil bacteria. It contains DNA. It contains genetic material. So from this BT, it contains a gene. This is called BT gene. BT gene. Its name is Cry1AC gene or Cry2BC gene. Cry1AC gene or Cry2BC gene. So this is Bacillus thuringiensis, soil bacteria. It contains DNA. Okay. So this is called BT gene. That is Cry1AC gene. From this BT gene, certain proteins are released. Certain proteins are released. And these proteins will act as endotoxins. The proteins act as endotoxins. These endotoxins kills some insects. Kills some insects like a bollworm. Kills some insects called bollworms. That means it is acting as bioinsecticide. It kills bollworm. That is acting as a bioinsecticide. So from BT cotton, Bacillus thuringiensis soil bacteria, a gene called BT gene or Cry1AC gene is there. From this Cry1AC gene, certain proteins are released, and these proteins acts as endotoxins. They fight against some insects, particularly against bollworms. Bollworms. In Telugu, it is a ladde purugu, senaga pacha purugu. Okay, bollworm. It kills bollworm. Thereby, it is acting as a bioinsecticide. Now, here by using rDNA technology, this BT gene is introduced into a cotton crop. Thereby, it becomes BT cotton. In a similar manner, BT brinjal also. Are you getting? So in this BT cotton or BT brinjal, you have to remember BT stands for Bacillus thuringiensis. This Bacillus thuringiensis is a soil bacterium, okay, in which a gene or a DNA is present, that is called BT gene. This BT gene is also called as Cry1AC gene, Cry1AC gene or Cry2BC gene, and this gene have been introduced into the cotton crop. Okay, so this gene is incorporated into the cotton crop. It grows along with the cotton crop. In the cotton, it releases certain proteins called as endotoxins. Are you getting? And these endotoxins are target specific. They kills only some insects like bollworm. They will not harm other insects. They are target specific. They specifically kills the bollworms which infest the cotton crop, which infest the brinjal crop. So in order to kill these bollworms, we have developed this BT cotton or BT brinjal. That BT is acting as bioinsecticide. Is it clear? So this is BT cotton and BT brinjal. BT cotton and BT brinjal. Got it? All right. What is the next one? Huh? Okay, we'll continue in the next class. Right. So tomorrow, in the first half an hour, I'll conclude this biotechnology part. So after completion of the biotechnology, then I'll proceed to space technology. Okay. So tomorrow, I would like to complete the space technology also, space. And after tomorrow, defense and some emerging technologies. Is it clear? Right.